Good evening, everybody. I welcome you all to the ninth session of live interaction sessions for the course Bioinformatics, Algorithms and Applications. So, I'm going to share my screen. Please let me know if my screen as well as the presentation is visible. There's an issue with my PowerPoint, so okay, it's opening now. I hope this is visible to you all. Yes. Please use the chat box as usual to interact with me. Okay, so shall we start the session? And there has already been a delay of about 12 minutes. Was the screen visible, the presentation? Just drop in a yes or no inside the chat box so I can start. Yes, thank you, Devamana. So, once again, welcome to the ninth week of Bioinformatics Algorithms and Applications. So, this week we'll be doing practice questions focusing on the topics protein folding rate 1 and 2, protein interactions part 1 and 2, and computer aided drug design part 1. So, we'll be looking into CAD in a greater detail in forthcoming classes, I believe. For this week, I have about 9 or 10 questions before going to our usual current assignment. So the first question is as follows: in fast folding proteins, which among the following is dominant? Option A, short and medium range contacts between polar residues. Option B, short and medium range contacts between non-polar residues. C, long range contacts between polar residues. And D, long range contacts between non-polar residues. What do you think the answer might be? Thank you, Devangana. There's one vote for A. What about others? Let me just check if I'm recording the session. All right. Any guesses? B, C, or D, or A? So, in protein folding, which contacts are important? Or which contacts determine the rate? You cannot say which is important and not, but if you're worried about the folding rate, thank you, Nagamani. So which type of contacts So short and medium range contacts will help the protein fold faster, right? Because they're able to find their contacting residues easily in close proximity, and therefore they can fold faster. But long range contacts take more time for the residues to come together and form interactions, right? So D or C will not help us. So amongst A and B, short and medium range contacts between polar residues, non-polar residues. So we know that non-polar residues are dominated in slow folding proteins, right? Therefore, we're going with option A as the answer, short and medium range contacts between polar residues. Fast folding proteins. Alright. Question 2. Which parameter shows a good correlation in all structural classes of proteins? Amino acid composition, long range order, contact index, helix parameter, and chain connectivity. Nagamani says A. Anybody else? Remember to observe this in the part of the question, structural classes. So what are the structural classes of proteins? At least in the lectures for this week, we had all alpha, all beta, and mixed, which has alpha plus beta, alpha, beta, right? So which parameter shows a good correlation in all structural classes of proteins? Is it amino acid composition, long range order, contact index helix parameter, or chain connectivity? For those who just joined, we are at the second question. Which parameter shows a good correlation on all structural classes of proteins? Last half a minute for people to attempt at answering this question. Alright. So it is long range order, right? So among all categories, you had a correlation between zero point 0.7 or zero point 0.8 for all structural classes, all alpha, all beta, or mixed. I don't remember the exact values of my head, but I think I did write it down somewhere. You can check the lectures, or I can include the values when I forced the PPT. But I thought this was a straightforward question for you all, given it was uh, repeatedly mentioned in the lectures. I will include the values for all alpha, all beta, and mix it. It was about 0.78, 0.80, and 0.81 for all of these, I guess. Right? So apart from that, if you look at other amino acid composition, it will vary depending on a protein is predominantly hydrophobic, or if it's a transmembrane helix, or if it's a beta barrel. The amino acid composition could vary quite differently between the all alpha, all beta, and mixed um, categories. But here we are not talking about the absolute values; it is the correlation, right? Obviously, amino acid composition will not be the same for all alpha and all beta uh, proteins. Similarly, long range order will be higher for all beta and not so high for all alpha. I mean, mixed classes, and quite low for all alpha proteins in general. But the correlation, what you predict and what actually occurs, the correlation between long range order and an all alpha protein or folding rate is higher than compared to chain connectivity or contact order amino acid, sorry, contact index helix parameter amino acid composition. Question 3. Which database provides the pictorial representation of non-covalent interactions in protein structures? Another straightforward question. Option A, PDB sum. Option B, PDB interactions. Option C, PDB TM. And option D, PIR. Again, last week I had pictures of expansions of each database. I don't think I included those this week. I will include in the answer page where, thank you Devangana. I'll put clips of the other databases so it is clear and it will be useful for you when you revise. Another straightforward question, right? So I'm not going to wait anymore for the answer. It's option A. Question 4. The energetic contribution due to main chain atoms and side chain atoms in protein complexes represent which types of interactions? A specific and specific, B specific and non-specific, C non-specific and specific, D non-specific and non-specific. So what are the interactions we're talking about? Main chain interaction, that is your A, or for, maybe I should do, thank you Devangana. Others, I don't see any participation. And thank you Nagamani. Side chain, side chain interaction. This is your second. So is side chain interaction specific? Yes, right? So that leaves you with two options, A and C. Nagamani and Devangana have already voted for C. So what's wrong with the first answer? So main chain interactions are not going to be specific, right? Because the backbone is almost the same for all amino acids. It's the side chains that give specificity. Therefore, non-specific is right. And option C is right. Clear? Okay. Question 5. Which of the following residues occurs predominantly in slow folding proteins: cysteine, asparagine, arginine, aspartic acid. Slow folding, remember that. Thank you, Devamana. Thank you, Nagamani. That's it. So, residues that are dominant in fast folding proteins are N, Q, K, S. Right? So that blows these options. And residues that are predominant in slow folding proteins are A, C, G, and L. How many do you find here? Cysteine. So arginine and aspartic acid are again charged polar, therefore will be helpful in faster folding. So slow folding, especially cysteine counts because it has to find, it might form double bonds and disulfide bridges. So in order to find the other residue and make that interaction takes time. Question 6. If the solvent accessibility of arginine 128 in ligand bound and ligand free structures are 34.4 angstrom square and 72.3 angstrom square respectively, comment on the location of arginine 128. So this is bound, this is free. Option A, it is at the interface. Option B, it is not at the interface. Option C, information is not sufficient. Option D, neighboring residue information is necessary to define the interface. Thank you, Nagmani. Okay, so This information is sufficient according to me. Therefore, I'm going to negate this option. The solvent accessibility surface area. So look at this. You have 34.4 angstrom square when it is in bound conformation. And when the ligand free structure, you have the same arginine 128 having 72.3 angstrom square. That's absolute, around double the value, right? So which means in ligand bound form, it has a lesser surface area because the ligand is touching. And the absent form, there's greater area for the solvent to access this residue, arginine 128, which means it is at the interface, correct? Had it been somewhere inside the protein, in the interior, this is arginine, and this is where the ligand binds. Okay, some other residues here. Let me give a different representation because these days it's not easy for me to change color. There's some problem with my pen. So if this were the ligand, then its binding or not binding will not change the solvent accessible surface area of arginine, right? So the moment arginine has a drastic difference in its accessible surface area in the bound and non-bound conformations, it means that it is at the interface, and not at the interface is wrong. Similarly, neighboring residue information is necessary to define the interface is not necessary. Therefore, the right answer, as Nagamani pointed out, is option A. Question 7. Protein folding rate is used to option A. Understand protein folding kinetics. Option B. Understand thermodynamics of protein folding. Option C. Predict protein 3D structures. Option D. Predict protein function. Thank you, Devangana. Okay, Nagamani says B. So we all agree that it's not C or D, right? I would argue it's not thermodynamics because in protein folding rate, we are not worried about bond energies or change in free energy or interaction and other aspects of protein folding, right? It is what the time required for protein to assume its native structure. So therefore, it is the more appropriate option would be to understand the protein folding kinetics. Okay. Question 8. Which of the following residue pairs is preferred at a heterodimeric protein-protein interface? Option A, aspartic acid arginine, cysteine arginine, glutamine and valine, alanine and valine. Thank you, Nagamani. Others? When you look into the preference for contacting residues, spargin and arginine have, I'm sorry, aspartic acid and arginine have a value of 8.64. Right? You don't find any other combinations as uh, a preferred pair. So all the others are wrong, and therefore spartate and arginine the right answer. Question 10. Which database contains the binding? I think I skipped question 9. It's already, we have more questions at the end of this year's assignment. Which database contains the binding affinity of protein-protein complexes upon mutation? So we have already seen Protherm, Pronet, PDB, some approximate is new, I guess. But again, for this answer, I did not include the databases because I thought we are all familiar, but I can include during uploading the slides. Thank you, Devana. Others? Without question, Protherm is for thermodynamic parameters. Pronet was for interaction between proteins and nucleic acids, right? PDB sum we already saw, pictorial representation of interactions. So proximate is the database that contains the binding affinity of protein-protein complexes upon mutation. Right. So this year's assignment, question 11, the binding mode provides information on A. Cavity in a protein, B. Location of a ligand binding site, C. Geometry of the ligand at the binding site, and D. All of the above. So from here, I'm expecting more answers to drop in the chat box because you're all familiar with the questions and answers. Thank you, Dimamino. Others? Okay, one vote for D. I mean, I was tempted by D as well, but the question I think is a very specific question to one of the slides in the lecture. So the binding mode provides information on cavity in a protein, location of the ligand binding site, which is kind of related. Mostly it's a cavity, or you might have a cleft where the ligand binds. Option C, geometry of the ligand at the binding site. It's a very interesting option or more attractive option. So definitely, I my heart goes for C. So, but when you're a little partial to this, that's when I remembered that it is a more directed question at this slide. Okay, so where you have the binding mode here, which concentrates on the geometry of the ligand at the binding site, location, orientation, and conformation. Whereas the binding site or active site, I don't know what mode is it is. I mean, there was pose and score. So binding mode. probably is supposed to focus on this option, whereas the location of the binding site and the cavity is falling in um, these lines, I believe. All right. So although I'm unable to, okay. So since Nagamani has reverted back to C, I think I can save my explanation. Okay. Question 12. Which of the following residues occurs predominantly in slow folding protein? So we just saw which residues were in slow and which were in fast. I hope you all remember what those were. So what are the answer? Alanine and leucine, asparagine and glutamine, asparagine and cysteine, serine and lysine. Lysine. Okay, now the miss is A. Dipamine also says A. So what do you have in slow? A, C, G, L. You have A, L, asparagine, glutamine. So you have C here, L here, E here. So we are going with option A. Question 13. Which of the following set of residues have a high propensity to be at the interface of heterodimeric protein-protein complexes? Same question, different options. Option A, alanine and glycine. Option B, methionine and glutamine. Option C, threonine and oops, glutamate, right? And arginine and tryptophan. Okay, Devangana and Nagamani say D. Any other options? Again, in references, I don't see any values for alanine and glycine. Methionine and glutamine, again nothing. Threonine and glutamate, nope. Arginine and tryptophan, 7.48, right? Yeah, I don't see any other combinations described as preferred or high propensity, right? So we are all going with option D. Question 14. Folding of a protein from its unfolded state usually takes option A, microseconds to milliseconds, B, nanoseconds to microseconds, C, femtoseconds to nanoseconds, D, attoseconds to femtoseconds. Devangana says A. And you look at the options, that seems, thank you Devangana, the most probable option, right? In nano to micro even looks kind of okay-ish, but femto to nano and atto to femto are I'm going to disregard. They'll be extremely fast, and I don't know if we have instruments accurate enough to even measure such. So in answers, I have three segments, mostly from Google. and this is from one research article, and this is from another research article. Um, this is, I did not put the DOI, um, but this is an article by my supervisor. The third one. So when you look into this, a quick inspection of this Oxford protein interaction database suggests that the most proteins fold on time scales on the order of millisecond with a median of 5 milliseconds. So that somewhere around milliseconds to microseconds is usual. The alpha helix is often considered the nucleation site, or alpha helices fold faster because they have short range of contacts, right? So they can immediately find or fold easily compared to beta sheets. That's been, emphasized throughout the lectures, or first step of protein folding, and the folding timescale for alpha helix. So note that it is not the entire protein we are talking about here, and alpha helix folding ranges from few nanoseconds to hundreds of nanoseconds. So for an entire protein, it's going to take a bit longer than nanoseconds, probably in microseconds range. So to further confirm, for some proteins it takes an hour to spontaneously fold into their biological active 3D structure, and we are talking about usual cases. There will always be outliers which are extremely fast and which are extremely slow, right? While others manage to do it in mere microseconds. The nine orders of magnitude range in observed folding times has usually been considered a manifestation of the large variability in structural patterns and amino acid sequences found in natural proteins. So, this nine orders of magnitude, microseconds to hour, but hours is not usual, right? The option is A. 15. What is the correlation between contact order and long range order? Option A, no relationship. B, negative. C, positive. And D, cannot be estimated. Thank you, Nagamani and Devamana. I think there aren't other people in the session, are there? No, right? Therefore, there's no point in waiting, right? Okay. So we are clear on that, right? Because contact order and long range order is just an extra parameter where you have um, a cutoff of, say, so many, the contacting residues should be, say, 12 uh, residues apart, correct? So therefore there will be a positive correlation between contact order and long range order. Long range order will be lesser than contact order, but greater the long range order, definitely the contact order will also be higher. So it is positively correlated and not negative. Of course, there is a relationship because long range order is falls under the uh, contact order parameter. I don't know how do you call it. So while contact order is the sequence separation divided by length into number of contacts, LRO is number of residues that form long range contacts divided by N. And negative is again round. Yeah, I'm emphasizing on this question because one of the students or people taking the exam had mailed me asking about this question, but I think he has not joined today's call. So if there are so many residues in contact, let's say the same residues, you will have contact order and LRO. LRO might be say 3 in so many 150 residues. You will definitely have uh, delta Sij of all the residues here within this cutoff region, summation of all those divided by length into contact order, and there's no way this can be negatively correlated. Negatively correlated should be if there's a rise in one, the other should go down, right? That cannot happen in contact order and long range order. And so more the long range interactions, contact order is high and folding rate is slow. So I have put this to justify the fact that greater the long range interactions, higher the contact order. I mean, even if you don't see the formula, when you think about the physical meaning, it makes sense, right? Okay, question 16. In an energy based approach, which interactions are used to identify the binding site residues in protein protein complexes? Option A, electrostatic and hydrophobic. B, electrostatic and disulfide bond. C, van der Waals and electrostatic. D, hydrogen bonds and van der Waals. Okay, I think I'm gonna say C. Again, it's a so does Devangana. So it's a straightforward answer, right? Van der Waals energy and electrostatic energy are the interactions used to identify binding site residues in energy based approach. Question 17. Computer aided drug design does not work on which of the following scenarios? When the ligand is known and the protein structure is known, known ligand and unknown protein structure, known protein and unknown ligand and unknown protein. Nagamani says D. Thank you, Devangana. Obviously, when you have both ligand and protein known, it is very easy to do. When you have no ligand, when ligand is unknown but protein is known, still it's possible. Protein is unknown, ligand is known, that's also fine. But when you don't have protein and ligand, you can. It's not uh, impossible, but it's not a same thing to do, right? Um, you're going against a lot of assumptions and variables. So when you don't know what protein you're working with or you're targeting, and you don't know what structure you're trying to bind with using a ligand, so it's a really blind approach to do anything. For example, let's go. You can use an analogy of a geographical location and the natural disaster that you can expect. So if you know if it's in Chennai, and you can predict what disaster might come in next an attack. Or if you know it's Bangalore, you can predict something. Or you know I'm looking for a cyclone. What are the regions in India that can affect? You look for the coastal regions or something. So I'm correlating the region with protein and disaster with ligand. Alright. So you will not expect a tornado to come somewhere here or a hailstorm in Tamil Nadu, right? So it's generally not expected. But when you don't know the region or what disaster you're looking for, then where do you start? Right? At least you know the place and you know the time and climate or which time of the year it is, you can predict something. Or you know what disaster you're looking for, then you can find out which regions are probable to get affected. But when you neither know what region you're going to work with nor which disaster you're looking for, it's a situation where computer aided drug design will not help you with unknown ligand or unknown protein structure combination. Okay, question 18. In Lipinski's rule of 5, the maximum log P is. Thank you, Divangana. Thank you, Nagamani. So the answer is 5. Not greater than 5, so it has to be less than or equal to 5. Therefore, the maximum will be 5. Question 19. Chevron plot is used to understand protein folding kinetics, understand thermodynamics of protein folding, predict protein three-dimensional structure, predict functions of protein. Same question, same options, just a different word. We have already seen this. That's right. Thank you, guys. That's right. Chevron plot is to understand the protein folding kinetics. Question 20. Protein A has a contact order of 8.7. Protein B has a contact order of 5.7. Which one folds fast? Devangana says B. Oh, Nagamani says A. Oh, that was for the previous question, sorry. So as contact order increases, Nagamani also agrees, folding rate will decrease. Right, so protein A has contact order of 8.7 and B has a contact order of 5.7. Since A has higher contact order, it will be slow and B should be fast. So I got three more questions, I think. How to predict protein folding rates from sequence? A. Predict contacts, LRO, and folding rate. B. Relate amino acid properties with folding rates. C. Relate properties, predicted secondary structure, and ASA with folding rates. D. All of the above. Nagamani says B. Devangana says A. Finally, something that you both don't agree on. So, predict contacts, LRO, folding rate. Is that true? Yes. So, how to predict protein folding rates from sequence? So, you start with amino acid sequence and go to predicting contacts and then calculate the LRO and then estimate folding rate. So with this in mind, A should look cool. Next, relate amino acid propensities with folding rates. C. Relate properties, predicted secondary structure, and ASA with folding rates. ASA is also, and predicting secondary structure and ASA falls under this process, right? Under the predicting contacts, how do you predict contacts from amino acid sequence when you don't have the structure? You will predict the secondary structure first, right? And also accessible surface area, you will set different cutoffs and model and predict the structure based on those models, and then predict the contacts. So before you predict contacts, there are a series of steps which are elaborated in C. Relate amino acid properties with folding rates. So this is a side point where again you are looking into different amino acid properties ranging from their accessible surface area, propensities, because how else do you predict the secondary structure, right? So therefore that is also included in vaguely, although not directly if you look into the steps this way, but everything is there in the background, right? So I'm going to go with option D, which is all of the above. Okay. Question 22. Protein folding rates provide the information on 3D structure from sequence, how fast or slow a protein can unfold, how fast it assumes its native structure, all of the above. Thank you, Nagamani. Thank you, Devananand. Uh, Amreen, so I don't know the exact solution for this. You can maybe send me your email address. So in case I get a solution for this, I'll be able to communicate with you. And please mute your mic in case it's unmute. So that's right. So you don't predict the 3D provide. There's no information on the 3D structure from sequence, right? So that crosses your option A and therefore B. So amongst B and C, protein folding rates provide information on how fast or slow a protein can unfold or how fast it assumes its native structure. So the appropriate answer would be C. Thank you, Amreen. I'll look into this and see how I can help. Did you try forgot password or something? I mean, I guess YM is working for all of us. Okay, so you should contact NPTL team, but I'll get the right address and pass it on to you. Okay, question 23, which is the most famous question of this week. Usually, which type of proteins fold faster? Thank you, Narumani. Thank you, Devangana. So with that, I can give this a rest, I believe. Oh, there's one more. Binding site residues refer to option A, residues at the interface. Option B, residues which are interacting between a pair of proteins. C, residues which are close to each other between a pair of proteins. D, all the above. Okay, now this is B. I don't remember whether I put in a justification picture for this. I had to do this week in a hurry, so plus my PPT crashed and it wouldn't open. Okay, Devangana says D. I don't think I've written down anything as well. But yes, I will definitely include some picture when I post this, because when we looked into this use of ASA as well as using distance, right? Those were the two methods that were discussed in lectures, if I'm not wrong, for determining which residues. Uh, so I've written here residues in a protein which are interacting with other molecules. But the two methods that we used were distance, accessible surface area, and then we had energy based approach, and four was binding propensity. It's a really bad handwriting. Right? So whenever we look into uh, the distance between two residues and having a cutoff, we don't actually worry whether they form a bond or interaction, right? Even when you have hydrophobic interactions or van der Waal interactions, there is always not a bond that really forms. So residues at the interface, definitely yes, true, which are interacting between a pair of proteins, again yes. So the option that generally would confuse you are is option C, which says residues which are close to each other between a pair of residues. Now, when you read close to each other, you might think so they are not really interacting, so it should not be a binding site residue, right? But when you determine with accessible surface area, even if it's close to each other, it is still going to affect uh, the accessibility for the solvent to come and interact with those residues, correct? Therefore, if you have a cutoff as if the accessible surface areas differs by just zero point 0.1 angstrom square as was described in the lectures, although that's extremely small, right? Even if you take some 15 or 20 angstrom square, it is still a good difference, and for, how do you say, reasonable enough for you to think that it is an interacting or binding site residue, because it is getting obstructed by another protein or another ligand or another uh, binding partner, which alters the accessibility or the energy or the distance or binding propensity values for those residues. Therefore, I'm going to go with all the above. Okay. Although you cannot actually call them as the active site where the substrate binds or interacts, binding site residue will still cover all of these cases. So this my questions end. I hope you found this useful. Thank you for joining today's session. I think the utterance is quite low this time. So there's nothing else left, thank you, Devangana and Nagami, for your continued participation as well as attendance. So if there's nothing else, I'm planning to Amreen. I will copy your email address so that I don't lose it. Okay, so I will get back to you. Yes. So that's it. Good evening. Thank you all. Meet you next week on Friday at 6 p.m. Okay.